right? But yeah, so what we do is downstream, and we are, so that's my wonderful group, that actually does all the work that I'm going to show you today. And we have two parts in the group, where one part is doing the method development and research, trying to make image analysis work for the more difficult cases, for new modalities, for the new samples, for the things which have not been solved before. And then we have the other part of the group where our software engineers are trying to actually bring these methods, and also the methods that other people develop, into the hands of biologists with or without computational expertise. Right, so if you've been following the image analysis world at all, then you know that since 11 years now, we find ourselves in the era of deep learning. Right, so this has been a big revolutionary push that happened in 2012. And there you can see that, so for example, for natural images, right, so this is already machine learning, right? So this growth, this is going from the explicitly defining what you do with images with having machine learning on top, right? Then this jump here in 2012, this is the first deep learning applications, and then you see here it actually finishes in 2018, but this is not finished, right? It looked like it plateaued, but then the transformer architectures were developed, and then it actually took off again. And this challenge now is almost saturated, right? And you can also, if you are as old as me, you could probably remember in the time when they just recognizing faces, not even touching who is in there, but just recognizing that a face is a face. There was a time when you would upload a selfie with a friend, right? And only one face would get recognized, right? And now we are actually there. So things have improved a lot. Can we now do everything? Not really. Also not for natural images, and also not for microscopy. So there's still a lot of very interesting things to look into. And if you generally want to ask yourself, so how is microscopy different from natural images? Can we not just, pretend like we have the same images like the people who detect faces and keep going? So there are a few important differences. One is that, so here are a few positive ones, such as that we don't have perspective geometry. This is actually great. Right? So our objects are always relatively to scale. It's much better than when you have to teach an algorithm that the car that is closer to you and the car that is further away from you is actually a car of the same size. Right? At the same time, we also can have real 3D. Right? We can have optical sectioning, we can have physical sectioning, but we don't try to recognize stereo from driving by a house. Right? And as the third thing, in fluorescence, we can even get targeted staining. Right? And this is a fantastic advantage compared to what you get in the natural image world, where most of this intelligence is going into learning one kind of foreground from other kinds of foreground, whereas we have the luxury of just looking at the foreground versus background problem. Well, there are also the negative sides, and these are that, well, most of these modern methods are actually supervised, and this supervision has to be done by biologists. Biologists are busy people. Their time is very expensive. Right? Because of that, there is very limited amounts of public data. Right? It's also, this data is not just that it takes expertise to annotate, it's also just hard to do. And what we have seen is this is an example from our collaborator, Alexei Mazel, in the University of Heidelberg. Right? So this was a 3D image stack of that size. Right? Annotating it took 4 months. Right? It's not 4 months full time, because you go crazy if you do it full time, but let's say 4 months in as much as you are ready to sit there and annotate membranes. Right? And then, if you're ready to invest this 4 months, then we can actually go pretty fast. But yeah, that time has to be put in. And then also, if you look at the tools that the community of the imaging scientists actually uses, you see that there is a very large prevalence of what we call point-and-click tools. Right? And these are, of course, very convenient, but that also means that for us, on the method development side, if you want to bring your tools to the people, if you want to have any impact on what is happening in the community, you have to actually wrap your things into the point-and-click tools. You can't just release code on GitHub. Right? So, well, Ilastik, that is now developed in my group, is one of the examples of such point-and-click tools. If you're not familiar with it, I thought I would still just show a quick video so that you would all know what it is that the group, I think, is most known for. Right? And so what Ilastik allows you to do is you take an image, right? You, with a brush, quickly label what things you want to have, what things you don't want to have, as many classes as you want. Well, people usually don't go above 16, but like here, it's just a foreground-background example. You can iterate with it, you can give feedback, you can see where things are going wrong. So that's the kind of things that we do. Right, so this is still not deep machine learning, right? This is a random forest. And we know that with all the advances that we had over the last 10 years, we actually came to the point as a community where for many modalities, not the most challenging ones, right? Not the ones where the signal to noise is so bad that you can barely see anything by eye, but let's say reasonable bread and butter kind of imaging, right? So there, automatic segmentation is now actually feasible, right? So if you provide enough training data, and if you know what you're doing when you're training, especially if it's a new problem, then we will be able to segment. This was not the state of things, say, 5 years ago when I was starting, when I was starting my lab, right? So I didn't, well, maybe I was overly pessimistic, but I didn't hope that we can catch up so fast. Right? But now it's indeed like this. So if you have sufficiently stable imaging system, you are ready to put in the time to provide ground truth, you can train an algorithm that will segment for you. But these two caveats here, I think, still remain kind of like roadblocks on the path to the adoption of these methods in the wider community. And we try to address them in my group. So, and also with the help of many other groups. So let's look at some of these things. Right? So how can we address the problem of never having enough training data? So we first encountered it when I just joined EMBL, and we joined this wonderful collaborative project of looking at 6-day sports fertilization worm called Platinaris dumerili, right? So it's a marine worm that was imaged with electron microscopy, like the complete thing. So the whole volume is about 8 terabytes, right? The animal volume is around 2. And well, we wanted to segment cells, right? And I did segment cells in EM before. The problem here, or let's say not the problem, but the exciting thing about this data set, that this is actually a whole animal, right? This is not pieces of the brain. So bigger data sets in terms of terabytes have been taken, right? But here, the big challenge is really that you have everything, right? You have all the cell types, you have all the tissue types. It's not all just neural. So when we came to it and we wanted to apply the usual approach that we have developed for segmenting neural tissue, well, so this usual approach, so that you know, is that you try to detect the boundaries between the cells, right? So the cell membranes. And then, because this prediction is never really perfect, you use these boundaries then to build up the real objects. Now, so we acquired some training data that was also around the brain, right? And then that's what we got. So we trained there, right? That was like a 30,000 euro investment into the annotation. And then what we get is a network that is actually not bad, right? So you see, I mean, this membrane predictions there, where it looks sort of like it did in the brain, it's doing okay. But then there are areas which it just cannot interpret, right? Like here, this edge of the muscle cell, it doesn't know what to do. So it doesn't find this membrane, right? So here you have a cavity, it doesn't find the membrane. And then there are many other locations like this. And the answer that the natural image community would give back to you is that, well, you need more training data, right? Your training data is not representative of the whole sample. The problem is that if you now wanted to segment the whole sample by hand, well, we only have one, right? So we would probably spend 3 years doing that, and then we don't need an algorithm anyways, right? So we started thinking, well, what can we do with that? How can we, without getting more training data, try to then still do with what we have? And in the end, we came up with a system that actually combined bottom-up and top-down cues. And there we, you know, on the one hand, we predicted these boundaries just bottom up, directly from pixels. But then we also put up the biological constraints, like the simpler kind, right? That a cell should only have one nucleus, you know, that a cell should not go over a tissue boundary. And then that actually helped mitigate the effects of some of these boundaries being missing. And then with all this, you know, after this method was developed, we could actually segment the whole thing. And now we know that it has 11,500 cells, right? And we know that they have a fantastic variety of morphology. Right, and this fantastic variety is why people actually take big data sets like this, but this is also what makes it so difficult to segment, that all of these things just look different as you go through the volume. Right, so it works for EM, right, it also works for light microscopy, right, so we then simultaneously applied it for some membrane-stained plant data, because we have great collaborators that work on plants, right, so you can take different plant organs with membrane staining, right, and see that, yes, they can all be segmented. And then we thought, well, okay, you know, it's all membrane staining. What if we look at something else that has membrane staining? Right, so then we looked together with Takashi Hiragi, who is now, I think, in Utrecht, was at EMBL at that time. We looked at 2 different mouse embryo segmentation problems, one at the very early stage, one around the peri-implantation stage. Right, so this is also somewhat challenging data, because as they need to be kept alive, the laser power cannot be too high. Right, so it is fairly noisy. But it still works, right? So the same approach of just, you know, getting these membrane labels and then seeing that if you have the additional nuclear stain, that you just don't merge the cells so that they have 2 nuclei, is actually working well, right? And yeah, am I pressing something wrong? Okay, let me try directly here. Oh no. Okay, let's see. Okay. And there is a tool that does all that. So if you need to segment images based on this kind of membrane staining data, especially if you have plants, because we have a lot of pre-trained networks for plants, I then go and maybe check it out. It's called PlantSeg. It's available on GitHub. It's you can use it with its own GUI. You can use it with Napari as a viewer. You can use it also in batch. Everything that we have trained is all available there. Right, so you can segment your own plants this way. Let's see if this thing works now. Yes, and that actually got us thinking then, well, that's good, but we still needed to label this, little bits of data sets that we use for training. We still needed to label densely. Can we label less densely? And that got us to the problem of positive unlabeled segmentation. And positive unlabeled is this regime in which you are not trying to take a small patch and then label really everything in it, but you just want to say, okay, I have this patch, and then here are a few objects, and then for the rest, I can't really say anything. I just, I know I can't say that this pixel, if it's not labeled, whether it belongs to foreground or background. Right, so this is known as the positive unlabeled setting. Right, and we found that this is what many of our collaborators actually want to have, because it's a real pain to segment images down to the last pixel. Right, so sometimes you as a human don't know, sometimes you just get tired. It also doesn't help because you don't sample enough of the variability across the different parts of the data. Right, so we thought that's an interesting problem to solve. The problem with it is, well, purely technical, that the selection of individual objects in training is not a differentiable procedure. Right? And let's not go into the technical details of why we need it to be differentiable. Let's just say that Adrian Volny in my lab actually found a way of how to make this object selection go with a soft mask, and now we can select individual objects in training, and we can also stabilize it if you only do very few objects. Right, and that, so the whole training procedure looks like this. If you're interested in the technical part, it's also published last year. Right, so you have some parts of the image labeled, some part unlabeled. So the parts that are labeled, you say, well, these parts should be as the human told you. Right, and the parts that are not labeled, you say, well, I don't really know what they should be like, but if you just like shake an image a little bit or you rotate it, then you should have the same result. Right? I don't know what it should be, but it should be the same. It turns out to be a very, very efficient constraint, and with that we can actually do things like this now. Right, so if you give us an image, like this one, where you label some of the things in there, right? Let's say all the ones that you could see, and then for the rest, you don't really know if there's still anything lurking in there, right? Or maybe you just already got tired. We could train on that, and then on the test data, we could actually get all of them out. And yeah, so that's a light microscopy example. We also have EM examples where we try to do it with, you don't have to do it from scratch, right? You can also train on something, then go to a new data set, and then when you do that on the new data set, you will find that it's underperforming because you have a big domain gap, right? And then you can just throw in this, say, 5% of the objects of the new data set, and then that would already adjust itself. So this is a Me2AM data set, and in here you can see like the part that I'm very happy about. It's like you can see these two things that look like tubes in cross-section. Right? And they are actually also mitochondria. So in this data set, there are some mitochondria that look like sausages on a stick. Right? And so if you cut through the stick, it doesn't look like the training data that was actually taken in the mouse brain, where you don't have this phenotype. And when we first applied a network that has only seen the mouse brain, right? So then we didn't have them, right? You see there's nothing here, and also the rest of them don't look so happy. Right? But then you throw in this 5% of the objects in the new data set, and you can actually recover them very nicely. Right, so that already got us happy, and then it turned out that for some reason this regime was not really explored for natural images either. Right, so we tried it also on street scenes, things like this. Right, so that got published last year, and now we are looking into how we can actually do it completely unsupervised. Right, so would it be enough to just have these consistency constraints, and how can we then make it run faster? Right, so that's still ongoing, but the part where if you're ready to label 1% of your objects, that's already there, already released. And yeah, so can we label even less densely? Right, so that was like the next thing that we got thinking about. And here, yeah, so can we do brush strokes? If you remember what ilastik works off, right, so these are all brush strokes. Right, we never have to paint the object completely. Right, so some of our users, also some of our developers actually say that I'm taking a brush that is way too thick. Right, so here it's done for demonstrating purposes, but you should actually take a brush that is like 1 pixel thick, because it doesn't need so many examples, right? It's nice, and it's gives you a faster turnaround time. So how could we get something like that, but better? Because that actually stops once your data becomes a little bit more complex than what I'm showing now. Right, so here, yeah, you see ilastik has been used for a ton of completely different applications we don't actually follow, right? We don't have any telemetry, so I could just randomly screenshot something. So it has been used for a lot of things. So clearly people appreciate this quick turnaround. And yeah, so it's not always good, but it should be good for something. And if you try again to come back to this mitochondria segmentation problem, and you train, well, let's say this is a particularly crappy random forest, but you see that it's actually learning something useful. Right? So here, like the mitochondria are there. There's also a lot of other things that it can't learn to distinguish, but the right things are all there. So then we thought, well, okay, maybe we can just learn to clean it up. Right? So we can put a network on top that would enhance the predictions of the bad classifier. And the cool thing about this network is that then it doesn't actually have to see the raw data. It just learns to clean up the bad predictions. So it only sees the bad predictions, and on the new data, you can then come, give your usual labels, get again the crappy predictions, and then never having to retrain this cleanup network, you can get much better predictions again. And then you can even use these predictions to train yet another network, and then that network would be really, really good. And here, so we tried on the, so I'm showing it on EM here because EM is like the most difficult segmentation problem. It also works for light. Right, so you can try it for mitochondria, right? You can try it for boundaries, right? It really brings something for free, right? Because the only labels you provide are the labels for the random forest. Right? You can even go cross modality because in the end, it only sees the probability map, right? So it doesn't actually matter if your original data is from electron or light microscopy or different kinds of light microscopy, as long as the objects you are looking for are the same size in pixels, approximately the same size. Right, you have to be careful because once you actually train it to find these structural priors, it will find them everywhere. Right? So if you are trying to enhance a different signal, it will still enhance whatever it is you trained it for the first time, like you can see here. Right? So here, if you wanted to enhance a boundary signal with a mitochondria enhancer, it will still find mitochondria. Right? They are not random, right? They are at the positions of the real things in the image. It just, they are still present at this kind of latent, somewhere lurking in the pixels of low intensity. Yeah, it also, why does it just turn off sometimes? Yeah, so let's see how this will work in ilastik. Right, this is already available in the beta version, and this is how we think this can be used. Right, so this is real time, but Dominic is driving it, so he's really, really fast. And yeah, so you see, this is again our little mitochondria problem here. Right, and we try to do it with a random forest. Right, and we find some areas that look like, yeah, these are good mitochondria examples. Right, you label foreground, you label background, and exactly like I said, you see that it's kind of, it figures it out, right? But not quite, not fully. It could get better, right? So then you can label some more, but now it, it thinks that other things are mitochondria which are not, right? So you could label more, but then, for instruction purposes, let's say that you are now tired. So you take a model that actually enhances this mitochondria predictions. It downloads itself, initializes itself, and now you can say that the mitochondrial signal is the one that you want to enhance. Right? And here you will then get improved predictions. Right? And then you can also label away the things that are actually background. Right? Then it looks at the probability map, they are gone. It will not create the fake things there. Right? And now you can zoom out, do it at the whole volume if you want. Right? You never need to retrain the network itself. Right? You just train the underlying random forests to steer it towards making better predictions. Yeah, so that is, like I said, available in beta. We are still experimenting with it ourselves, right? And we also need to train all these enhancer networks. So that's, like, we have a lot of hope for this technology. Try it out if that's the kind of problems that you want to solve. But like I said, we are still really working on this. Right, so these are some of the kind of the research things that we've been working on. And now I wanted to switch gears a little bit and talk about how can we bring these methods to people who are not experts in deep learning. Right? Or how can we get rid of this second roadblock of like that the training has to be performed by an expert. Right? So of course there are models out there that have been trained so many times that also as a non-expert you could probably do it. If you have a new problem, that becomes a little bit more tricky, but for old ones, I think it's getting easier and easier. But in general, there are a lot of people that train networks, especially for natural images, but also for microscopy. Right? There are very big conferences where people mostly submit deep learning work. Right? And then it's all there. Some would then be accessible as code, for the good people that actually submit code. Very few would be accessible as tools. And then if we ask ourselves, how can we now bring it to the people that would actually profit from them? Right? And then remembering that many of the imaging scientists don't code. Right? Many don't deeply understand deep learning and probably shouldn't. Right? They have other things they need to understand. Right? And yeah, and even for annotations, we don't have enough tools. So maybe putting all the work into developing a whole new set of tools is also not the most practical thing. Right? So yeah, we got together with a few other very nice people from the community, and we started building the BioImage Model Zoo, right? Which is the place where we can share the models and everything that is related to models. And here you can see the partner softwares, right, including ilastik. Right? So all these people are actually actively participating in making the zoo what it is now. Right? And the main thing about it is that we have managed to arrive to a metadata description of the models that allows different tools to run the same model. Right? And that means that for me, as the developer of ilastik, I can hope that people in other labs will submit models there, and then our users can just run them. And then conversely, when we develop stuff, right, and then people want to run it in ImageJ because they prefer ImageJ, that's fine by me, right? Be my guests. They can then take these models and run them in deep ImageJ, and it will still be fine. Right? And so here you can see that if you, for example, if you don't want to code, right, you can take the Stardust model from the zoo, run it in deep ImageJ, do things, right? You can run it in QPath, do the curation in there. QPath has fantastic curation mechanisms. If you actually don't mind coding, you can go back to the notebook where it was trained and change things there. Or you can go directly to the Stardust Python library and use the network in there. Obviously, you can also use it in ilastik. And so here you can see an example of one of these plant networks that I was talking about before that actually allow you to find the cell membranes in plants. And yeah, so there you would have the prediction, and then there is a two-click operation in ilastik that will bring you from this boundaries to the actual objects. Right, and so there's a preprint about it, and you can see there are a lot of people involved. Some of them are actually here, right? And it also, the list keeps growing, right? So we put out the first version of the preprint last year, and then other people wanted to join, and then we realized there's also other things missing. So the author list is growing, and then it's also not too late to join in if you wanted to make a major contribution to the zoo, or then we will have probably the second iteration again. So yeah, then once we had the prototype, we also applied for funding with it, and we got a grant from the EC to establish a consortium that we call AI for Life. Right? There are 10 partners in there for 4 million euros who will now, over the course of 3 years, try to make the zoo out of a prototype into an actually useful tool for the community. And we just celebrated 1 year, so I wanted to quickly tell you maybe a little bit of the things that we've been up to. Not all of them are fully, production ready yet, but a lot of them can be played with already. Right? So yeah, the main thing that actually makes the zoo work are the standards and libraries that we are developing. Right? And so the standards are what allows the different tools to run the same network without anyone having to look into the code and readjust the code every time. Right? And so the metadata of the networks has been developed in over 3 years of consultations and clashing opinions and trying to bring people forward. So that was an interesting experience in itself, but I think we now have a nice process. And so you can specify pre-processing, post-processing, input shapes, output shapes, a few of the other important things, the authors, how to cite it, all of those things. And importantly, so there is the specification of the resource that you can give, but then there are also a library in Python and a library in Java that would allow you to then do all these operations that we are doing in our tools. So if you have a tool of your own and you want to run the networks that have already been uploaded to the zoo, then there are these very helpful libraries in Python and in Java that would allow you to do it in no time at all. Yeah, and that actually, the presence of this library, we hope puts us on the way towards a reproducible system, where of course, to do something like this, you would have to code, but that means that you could, in your code, take the networks that were submitted for challenges, in this case, the Human Protein Atlas Cell Classification Challenge, and reproduce them yourself, right? And this is very easy because you just read them, they are in the right format, you can read, you can run inference. So yeah, you can reproduce everything that they have done. With a custom Python code, and then get to the segmented and classified cells and enjoy. Yeah, so all you need to know about a model to be able to run it programmatically is actually in the model card. And then the most, let's say, disciplined developers actually submit everything. So the pre-trained model, the data that it was trained on, and also the notebooks that were used for training it, like you can see in the example here. And they are not all like this because we actually wanted to be inclusive, but we are trying to maintain that as a standard. Right? And then, if you have all this information, we also started thinking, in particular Wei Yang, that is actually a lot of quite rich things in there, especially if there is also the documentation on how the model can be run, and all of this is referenced, right? And that's how the BioImage chatbot has been developed, right? And that's a thing that just got released, I think, a week ago, right? And there it's based on GPT-4, and it can give you advice about bioimage analysis and about the correct use of the zoo. And Wei and his co-authors are really looking for beta testers of the tool. So if you go to this GitHub repository that I have pointed out there, and there will be a form how you can sign up. So if you like playing with large language models and like playing with this kind of coding assistants and other kind of assistant GPTs, please go there and help Wei make this tool actually useful. Right, so another thing that we hope that this kind of a zoo of pre-trained models can do is give the horrible process of data annotation a bit of a boost. Right, so if you have a network that was trained on data that is not exactly like yours, but somewhat similar, right, so that you can get to, say, 80% of the way, then correcting these errors usually takes a lot less time than trying to segment from scratch. Right, so if you now choose a network in here, right, and then put it into ilastik, and out of these boundary predictions that were not perfect, correct a few so that they actually become better, you now have new ground truth data. On this new ground truth data, you can go to the zero-cost DL from MIC project and then fine-tune the model because you now have new labels, right? And then, first of all, upload the model back to the Model Zoo because you now have a new model that works on data that it didn't work before. And then you can go, for example, to DeepImageJ and start to analyze the morphology, right? And the reason why I mentioned fine-tuning here, and well, that's zero cost as the organization are a partner of the Model Zoo, precisely for that, right? So that we could have reproducible ways of how to fine-tune networks, and so that people could also have examples of how these networks can be trained. And then the team behind the zero cost has now, I think, also last week released the new version of it that is called DL from MIC Everywhere. Right? And where they are also really asking for beta testers to come and try it out and tell them how it's working for them and is it actually what the community is looking for. So if you like trying things out, please also go to these guys and try out their notebooks that are now not limited to Google Colab, but actually through containerization can run anywhere on your institute cluster, on your local machine. Right? And this way we hope that we would then have an even better set of methods that we can recommend the people for training if they are not themselves deep learning experts. Then BioImage Archive is also part of the AI for Life initiative, and they are participating in the sense that they want to, well, in general, their mandate is to collect all the published bioimaging data. Right? And with us, they actually want to make as much of this data as possible AI ready. And they already have a little collection of AI data sets, and I think they are very encouraged by reusability of this data because these data sets are not big and people that need to train something often go there. So actually these AI-ready data sets are some of the most used ones from the whole BioImage archive. So submit yours if you have annotations, right? This is great. People will find them very useful. And here you can just see an example of the geometrical annotations. And so as you probably know, there is no single standard of how you can save annotations. And this is also part of the things that they are now trying to establish together with the team behind OMEZAR, right? So if we have biological data, how can we save the annotations in one way and not in 10,000 different ways? Right now, for the people who think that they need more help with their image analysis, but also for our side to make sure that we are not missing some of the big problems that the community is facing, right? And we don't have everyone on the computer science side working on nuclear segmentation while people are suffering without something else being solved. We also run open calls. And so the first one ran this spring, finished March 31st. And here, we tried to reach out to as wide a community of imaging people as we could. If you haven't heard about it, then probably we didn't reach as widely as we should have. There will be another call next spring. So if you have a problem that you think, if it were solved, then it would really push your biology community forward, we would really love to hear from you. Right, so in this very first call, we had 72 applications, right, that they were then assessed by a large panel, and there was a complex process involving many different optimization routines trying to select the 10 problems that we actually now are solving. Right, so here you can see some of the results of, the kind of things that we wanted to find out, right? So for all these outstanding problems, how big are these images? What kind of modality are they in? Are they 2D? Are they 3D? So for me, for example, I was surprised that 2D is still such a big problem. Because for me, usually people come with 3D problems, and that's really such a pain. But no, 2D is also still absolutely not solved. There are many problems on the 2D images that we should still be addressing, and now we will address at least some of them. If you have other ones, please apply when we'll have the next call. Now, coming to the end, to summarize, so my group works a lot on the segmentation methods, and we now see that the modern segmentation methods, not just through our work, of course, but through the work of the whole very big community that works on microscopy, but also the very, very big community that works on natural images, that these methods have now become very powerful, but the training annotations remain a major bottleneck. So we had some ideas on how this can be alleviated. We will keep working on this, but we would also be happy if you want to reach out and try some of the things out. And what I think is coming is, well, we see that there is a big momentum in people wanting to share the pre-trained models, and also people being eager to use pre-trained models. So I hope that now, as the next on research question, we can look very seriously into fine-tuning, and adaptation of networks and see what can be done there. This is surprisingly understudied, because usually people just say, well, fine-tune on some data. And then, exactly on what and how do you select these areas where you fine tune, and what parameters do you set, and how do you make it so that every biologist can use it? Not really done yet. So, and I find these are all very, very interesting questions. So I'm hoping for a big rise in deep learning used by non-experts, because things just become a lot more accessible. But it also means that on our side of the community, we have to be ready to talk about the pitfalls and the downsides, and how you can validate, and how can you make sure that you can trust the results. And sometimes we also don't know ourselves, so this has to be an active dialogue, and we have to think about this also, not only me as a deep learning expert, but also, all the domain experts, because I don't know how to validate these things. I don't know what kind of results you expect. So one has to put some thought into how can you make sure that the results of the networks are actually trustable. But I think this is now a great time to start thinking about this. And then finally, please share your data, because this is exactly what we need to train more models and to bring this technology further. Thank you very much.
Okay, maybe I'll start with one. So the BioImage Model Zoo looks really, really amazing. So if I understand correctly, I don't need to install anything, and I don't need to download anything to use it. Is it correct? Or
well, it depends on what you want to do with the models, right? So the idea was that you have already your own favorite tools where you like to analyze images, such as Fiji, right, or Elastic, or maybe IC, right, or maybe QPath, and then you don't need to download anything else, right? So into these tools, you can find yourself a network in the zoo, and then you can use them in the tools that you're already familiar with.
Okay, and the model card you showed, this I don't need to download neither. I just need to...
No, so the model cards are there for you to choose the models, right? So this is like a bit of, like when you go online shopping, right? And then they are there, and then you can click on them and look at details and look what data they've been run on before. And so you do all that. And then in the end, you click download because you need the model locally, right? And then it downloads, and you can, on a small test image, you can actually run out there, right? So, but this is really just like a sanity check, right? So, and we also do these checks ourselves, right? So we try to make sure that all the models that are in the collection, at least on their own testing data, produce the right results.
Okay, thank you.
Hi, the synergy between the DL and the annotation, manual annotation is to me a very good idea. I was wondering, do you plan in the future also to add some, I would say, geometrical segmentation that biologists are used to use several times, even if it's not perfect? Sometimes it's, I would say, partial and partial enough to feed your elastic. So do you think it would be feasible?
I think everything in the end can be combined, right? So if your problem is such that it can be addressed with geometrical segmentation, right? If you're, I don't know, looking for circles, right? And then, sure. But that was kind of what we tried to achieve there with the Model Zoo, right? That you can just put deep learning as an add-on then to the other tools that you have, right? And in the way how we have used it as a prediction enhancer for the, for the cases when the problem is too hard for elastic, right? You can put deep learning on top and make the previous steps better, Right? So I hope that in a similar way, we will also be able to combine it with the other kinds of analysis that people were using before.
Any other questions?
Hi, just one question. I'm using that also for calcium imaging, and it's a kind of image when you can do simulation very, very easily. And what has been shown for a long time and in several papers is that if you ask humans to annotate, they systematically remove some type of events. Any ideas to how to remove the human bias, basically? Because we really like falsify the set of data by doing that.
That's a very interesting question, right? And I think if you need to have an alternative way to get to this ground truth, right? So we usually call the human annotations ground truth, right? But they are not always, let's say, the golden standard, right? And like you said, in this case, I think if you can have a different modality or a simulation that will actually be more truthful, this is of course even better, right? So if you can, sort of like if you think back to the KRE algorithm, right, for image restoration. If you can fix your sample and use that as training data, right, that's the best, right? Because this is true. Right? And at the same time, like the humans are not perfect. So we have to then think about how can we get the better ground truth that is not, if you know where the humans are usually making the systematic errors, right? You can make a network look at this harder, right? Or you can discard these areas somehow, right? So maybe, I don't know how the simulation algorithms run, right? But maybe if one could add on top something that where that humans would miss. And so usually just playing with the training data does the trick, right? But they are very, very dependent on the training data. If you fit in something that has a certain bias, it will completely reproduce this bias. Right? So having a few human errors is okay, as long as they're not systematic. Right? If it becomes systematic, then yes, all of this will be missed.
Any other questions? We have time for one more question.
So if I'm not mistaken, I mean I have the impression that most of the work on machine learning in segmentation issues is done in space, in the spatial domain.
Yeah.
But as a matter of fact, why not? You could take 3 or even 4 dimensions and include time in the whole thing. And one could think about, for example, the localization or the identification of certain events. Somebody mentioned, for example, calcium data as a segmentation problem in space-time. Do you know if there's any work going on in that sense?
Yes. So, so first of all, if you want to segment 3D data set that has a time dimension, right, there are ways to incorporate time into segmentation. Right? So the simplest way is to pretend like your time frames before and after are additional channels, right, and pass them in as channels, right? So that already usually brings the most improvement. Right? There are also other ways in which you can, presenting multiple of these time points, put in additional constraints like, the segmentation between the 2 time points should not be too different and so on. There are also other ways that maybe are doing it. So you can also, if you have 2D plus time images, right, you can interpret them directly as 3D, right, and then just do convolutions in 3D and pretend like time is just another spatial dimension. Right? That also, depending on your time resolution, can work very well. Right? And that is a popular approach. The problem is if you have 3D plus time, right? Because 4D convolutions are very slow. Right? So there, either you do this kind of consistency, or what people have also done before is you can do, so that's usually done for tracking, right? But what you can do is you can do like this hypothesis-based tracking when you have multiple segmentation hypotheses in every volume, right? Where some of them could, for example, put in the things that are like low intensity and a human would likely miss, right? And then you try to incorporate the constraints that would come from tracking, such as that, time should only go forward, right? Cells should divide and not compress themselves. And then that also really helps to clean it up and to find the one segmentation solution that is actually there. Yeah, so I think it's, there are ways to do it also because natural video is a very popular modality. But 3D plus time is still a bit tricky just because of the enormous data amounts in this.